Today, I will provide our overview of our research of transposon-based CAR T cells, with a particular focusing on how we can improve the function of CAR T cells. So in this morning, we had a very special lecture about the basic concept of CAR T cell therapy, so that I can start with the mechanism of the function of CAR T cells. So basically, one crucial aspect of the CAR T cell therapy is that the differentiation status of CAR T cells can affect the functions. The stem cell memory-like CAR T cells are correlated with the long-term functionality that have a high proliferation capacity and longevity in the body. On the other hand, the effector-like T cells display a rapid cytotoxicity, but lose their function quickly due to the exhaustion. And it's important to note that the manufacturing process significantly impacts CAR T cell differentiation. And this is a very good example that clearly demonstrated the phenotype of CAR T cells determine the clinical responses. In this clinical trial of CD19 CAR T cells for the patient with the chronic lymphoblastic leukemia, the researchers divided the patient into two groups, the responders and non-responders, and they tried to find what is the determinant of the clinical responses of these CAR T cells. And actually, there are no big differences in terms of the disease status or the biology of the tumor cells, but they found the clear differences in the phenotype of the CAR T cells in the final product. And the CAR T cells product established from the responders showed a memory-rich phenotype with less expression of PD-1. And the researchers concluded that the phenotype and the presence of the early T cell exhaustion in CAR T cell product determines the clinical responses. So to enhance the CAR T cell functions, the various modifications as listed here have been explored to produce memory-rich, less exhausted CAR T cells. And in these modifications, our team have been extensively researching the piggyBac transposon-mediated CAR T cell manufacturing, not only its simplicity and cost effectiveness, but also its the favorable characteristics. So back to over 10 years ago, our team leader, Dr. Nakazawa, first started to apply piggyBac transposon system for the genetic modification of T cells. In this study, he introduced GFP and delta CD19 genes into T cells by piggyBac transposon. And these cells were stimulated and expanded with anti-CD3, CD28 antibodies and IL-2 supplementations. And piggyBac transposon effectively introduced the gene of interest into T cells, but the transduction efficiency should be improved, and the purification step was needed to obtain the high population of gene-modified T cells. Moreover, as you can see here, that these T cells showed a CD62 negative, CD28 negative effector-like phenotype. And this is the first report of piggyBac transposon-mediated CAR T cells targeting HER2 molecule. In this study, the piggyBac HER2 transgene was introduced into T cells, and these cells were expanded with EVV-LCL and cytokines to generate piggyBac HER2 expressing CTL. And approximately the 40 to 50% of CAR-positive T cells showed CD62 ligand positive, CD45 allo positive central memory phenotype, as you can see here. And even though these T cells require the purification step to obtain the high population of CAR-positive T cells, the piggyBac HER2 CAR T cells effectively debulk the glioblastoma xenograft model, as you can see here. So as you know, well, the retroviral CAR T cell production involves T cell stimulation in the initial step because of the nature of the viral transduction that the retroviruses can only infect activated and proliferated T cells. However, this step leads to the strong T cell differentiation. Indeed, in this study, a conventional pre-activation step by CD3, CD28 antibodies for manufacturing of retroviral CD19 CAR T cells reduced the number of stem cell memory-lagged T cell fraction in the final product. So indeed, the excess activation of T cells could lead to T cell differentiation and early T cell exhaustion. So we have established the strategies to produce piggyBac-based CAR T cells without inducing the excess T cell activation during the manufacturing step. We avoid the pre-activation step. We just use the resting T cells to prevent the T cell exhaustion caused by the intense activation by the CD3 antibodies. Instead of using hyperactive transposase, which could lead to excess integration into the genome, we chose the original piggyBac transposase to ensure the controlled copy integration copies. For the optimal transfection efficiency, we carefully optimized the electroporation process, and we also optimized the expansion step using antigen presenting fetal cells rather than CD3 antibodies. And also to support the expansion of electroporated T cells, we supplemented the culture with IL-7 and IL-15. So why do we follow this manufacturing step? This is a paper describing about the manufacturing of piggyBac CD19 CAR T cells in our group for the first time. In this study, we generated piggyBac transposon-mediated CD19 CAR T cells, and these cells were stimulated by anti-CD3, CD28 antibodies for expansion. However, as you can see here, these cells showed massive cell death by the first stimulation, probably due to the activation-induced cell death, and required the selection of CAR-positive T cells. Therefore, we avoid to use the antibodies for the expansion of CAR T cells, and instead, we used the viral peptide pulse PBMC as feeder cells, just like the concept of the EBV-specific CAR T cell manufacturing. So in this study, we made PBMC-derived feeder cells that are pulsed with EBV, CMV, and adenovirus peptide, and piggyBac CD19 CAR T cells were stimulated with these feeder cells. So we successfully expanded CD19 CAR T cells with excellent efficacy. However, the final product did not show the specificity of these viruses, as evidenced by the ELISpot assay, as you can see here. And moreover, when we depleted CD19 positive B cells from feeder cells, these feeder cells failed to expand CD19 CAR T cells, as you can see on the right. Furthermore, when we attempted to use this protocol to generate CAR T cells targeting solid tumors other than CD19 target, the results were not as successful. Here we tried to make piggyBac HER2 CAR T cells just by following the previously established protocol for piggyBac CD19 CAR T cells. We could not see any HER2 CAR positive T cells, as you can see here. So, in manufacturing of the piggyBac CD19 CAR T cells with PBMC-based feeder cells, the CD19 molecule on B cells gives a CAR stimulation as signal 1, and the co-stimulatory molecule expressed on B cells would mimic the physiological immunologic synapse that can lead full activation of and expansion of CD19 CAR T cells with a physiological tone, not excessive as CD3 antibodies. Therefore, we hypothesize that the genetically modified antigen-presenting fetal cells that can express modified antigens and ample co-stimulation might mimic the physiological immunologic synapse, as you can see here, that can provide the optimal activation of T cells. So to do this, using HER2 CAR T cells as a model, we developed autologous PBMC-derived fetal cells that overexpressing the modified antigen and recombinant T cell activators, and we stimulated the CAR-transduced T cells with these fetal cells. So we have tried to establish piggyBac CAR T cells targeting HER2. So initially, we divided PBMC, one for CAR T cell manufacturing and the other for fetal cells. We overexpressed truncated HER2 and T cell activators, in this case co-stimulators, on fetal cells. And we have optimized the electroporation protocol of DNase transposon plasmid into non-activated resting T cells. And these electroporated T cells were then mixed with the fetal cells on day 1 for further expansion. Finally, we could obtain around 70% of CAR-positive T cells with dominant CD8 positive and stem cell memory-like phenotype defined as CD45LA, CCL7, CD28, and CD95 positive fractions. And notably, these T cells have scarce PD-1 and low LAG3, but high TIM3 expression, as you can see here. Obviously, to maintain the memory phenotype of CAR T cells, co-stimulation would be very important. Actually, we stimulated CAR T cells by PBMC-derived fetal cells expressing only the modified antigens or together with the ample co-stimulation. And as you can see here, the fetal cells expressing modified antigen and co-stimulation greatly induce the T stem cell memory-like phenotype than the fetal cells expressing modified antigen alone. So this suggests that the necessity of CAR T cell stimulation through the immunologic synapse, not only with the antigen CAR complex as signal 1, but ample co-stimulation. And this is the in vivo data of HER2 CAR T cells that control tumor growth. And as you can see here, the piggyBac HER2 CAR T cells showed excellent anti-tumor efficacy in vivo. And furthermore, when the fresh tumor cells were inoculated on day 28 to these mice, this tumor was totally rejected in the mice treated HER2 CAR T cells, suggesting the long-term functionality in vivo of the CAR T cells that can reflect the phenotype of the CAR T cells. We also wanted to see the direct comparison of the potency of the retrovirally or piggyBac transposon-based HER2 CAR T cells. We generated the retrovirally transduced CAR T cells and piggyBac HER2 CAR T cells, both of which has the same CAR positivity around 20%. Here is the real-time cell analyzer data, and the y-axis shows the number of tumor growth, as you can see on the magenta line. When CAR T cells were added to the tumor cells, the number of live tumor cells were decreased, as you can see here, and notably, the piggyBac HER2 CAR T cells demonstrated the more prominent and durable tumor control compared to the retro CAR T cells. Okay, so we have extensively investigated piggyBac HER2 CAR T cells with another target, including the CD19, GD2, EphB4, and FLT3, and so on. And we got consistent result with a high population of stem cell-like memory phenotype with CD45 allele plus and CCL7 plus fraction. But as you can see here, we generated these memory T cells from just around 30% of CD45 allele positive memory PBMCs. So why our CAR T cells enriched by CD45 outlet positive memory-like T cells. To answer this, we divided a PBMC into CD45 outlet plus or minus PBMC, and we made piggyBac CD19 CAR T cells from these two fractions. This is the CAR transgene expression on resting T cells 24 hours after electroporation. And as you can see here on the right, the outlet positive PBMC showed the much greater transduction efficiency than outlet negative PBMC. Then we cultured the T cells for 14 days to make the CD19 CAR T cells, and we checked the phenotype. Later on, I will mention this as outlet plus CAR and outlet minus CAR. And as you can see on the top, in outlet plus CAR T cells, we can see more significant expansion and higher CAR positive T cells with a memory phenotype and CD8 dominant fractions, lower PD-1 expression, and lower expression of CD57, that is T cell senescence markers, than in outlet minus CAR T cells, as you can see in the bottom. We checked the immune exhaustion profiles of outlet plus or outlet minus CD19 CAR T cells with or without antigen stimulation by transcriptome analysis. And as you can see here, the before stimulation, outlet plus CAR T cells showed less expression of canonical exhaustion markers as highlighted in blue, and these exhaustion profiles even maintained after the antigen stimulation, as you can see on the right. So we performed the in vivo stress test in which CAR T cell dosage was lowered to the functional limit so that these CAR T cells should be maintained and expanded in vivo to achieve the anti-tumor efficacy. And as you can see here, outlet plus CAR T cell groups showed better tumor control. So we evaluated the expansion of CAR T cells in vivo by a sequential bone marrow aspiration of the mice. And in the two long-lived mice treated with outlet plus CAR T cells, the CAR T cells were expanded even day 50 after treatment, reflecting the characteristics of the memory-like phenotype. So based on these findings, the quality of the starting material influences on the manufacturing and potency of piggyBac CAR T cells. And the selection of CD45 outlet positive PBMC may have a potential to produce more stable manufacturing and the favorable efficacy of piggyBac CAR T cells. So in summary, the piggyBac CAR T cells have appealing characteristics as their simple manufacturing process and the favorable memory-like phenotype. And the memory-rich piggyBac CAR T cells possesses durable anti-tumor potency, and piggyBac-based CAR transgene is likely to be introduced into outlet plus PBMC, and these cells were dominantly expanded, maintaining their memory phenotype. Okay, let's move on to another topic, how we can modify the magnitude of T cell activation to maintain their memory and unexhausted phenotype for the cancer treatment. So as we learned in the morning lecture, the signaling pathway of T cells can be explained in these three signals. The signal one is the T cell receptor signaling induced by the antigen stimulation, and the following phosphorylation of the cytoplasmic lesion of the CD3 zeta chain. And this signal one is further enhanced by signal 2 co-stimulation and signal 3 cytokine and chemokine signals. And furthermore, the excess activation of signal 1 also activates the immune checkpoint pathway, including PD-1, that leads to the strong deactivation of the T cell signaling and exhaustions. To control the CAR T cell exhaustion caused by excess activation of the T cell signaling, we have tried to control T cell activation by modifying the downstream of the T cell signaling. And we are interested in using RAF-MAP kinase inhibitor trametinib with GD2 CAR T cells against neuroblastoma, because basically I am a pediatrician and my clinical interest is the neuroblastoma. And also the relapsed neuroblastoma cells frequently acquire RAF-MAP kinase pathway mutations, so that the MEK inhibitor can be useful for treating neuroblastoma directly, and the combination of the trametinib with CAR T cells would synergize the function of CAR T cells. Okay, this is the real-time cell analyzer data that showed the proliferation of the neuroblastoma cells co-cultured with GD2 CAR T cells in the presence of trametinib. The tumor cells grow in the analyzer, as you can see in the red line, and when the GD2 CAR T cells were added to the analyzer, the GD2 CAR T cells kill the neuroblastoma cells immediately. And when trametinib was added to the co-culture, trametinib immediately controlled the function of GD2 CAR T cells, as you can see here. The GD2 CAR T cells were strongly activated by the stimulation, confirmed by a strong phosphorylation of the ARC1-2 proteins, and the trametinib effectively blocked the induction of PARC and the induction of the T cell exhaustion markers, including the PD-1, TIM3, LAG3 was also blocked. By contrast, the proliferation of the CAR T cell induced by antigen stimulation was not impaired by trametinib, probably because the main signaling pathway of T cell proliferation would be not signal 1, but signal 2, and would not be fully blocked by trametinib. So we hypothesized that the controlled activation of GD2 CAR T cells by trametinib would enhance in vivo function of GD2 CAR T cells. So in this case, we inoculated the neuroblastoma cells that did not have RAF-MAP kinase pathway, RAF-MAP pathway mutation, so that these cells were basically resistant to trametinib. And we gave the vehicle, trametinib, and GD2 or both. So in this experiment, we gave just 2 million of GD2 CAR T cells, which is a relatively low number in this kind of experimental setting. So, where's the point? Okay. So the anti-tumor efficacy was modest in the CAR T cell treatment group. But as you can see here, the CAR T cell with trametinib showed better tumor control, and these CAR T cells were long-lived in the peripheral blood of the mice when treated with trametinib. So indeed, the transient inhibition of CAR signaling can modulate the excessive T cell activation. In this paper, the researchers used dasatinib instead of trametinib to modify the T cell activation. And they clearly showed that the exhaustion in CAR T cells is promoted by excessive CAR signaling, and the transient inhibition of CAR signaling by dasatinib reverses the phenotypic hallmarks of exhaustion by remodeling transcriptionally or epigenetically, and that restores the anti-tumor functionality of CAR T cells. We have also tried to improve the CAR T cell function by the inhibition of JAK2 pathway. Initially, we wanted to treat the PH-like ALL, that is a pediatric type of ALL having a worse prognosis and basically the chemo-resistant, but can be treated by CD19-based therapy. Because these PH-like leukemia cells frequently acquire JAK2 mutations, and clinically available JAK1/2 inhibitor ruxolitinib can be effective and has been tested in the clinical setting, we thought that the combination of JAK pathway inhibitor with CD19 CAR T-cell therapy would have some additive effect. Here is the schema of the T-cell activation system, and JAK1 is the key molecule of the T-cell pathway or various cytokine system. Therefore, we hypothesize that use of the ruxolitinib, that is a JAK1/2 inhibitor, would impair the function of CAR T-cells. To see the effect of JAK pathway inhibitor on CAR T-cells, we performed the sequential co-culture assay using B-cell leukemia cell lines without JAK pathway mutations with CD19 CAR T-cells. We co-cultured these leukemia cell lines with CD19 CAR T-cells for 3 days. Then we add fresh tumor cells for the second round and cultured for 3 days, and similarly for the third round. As you can see here, the CD19 CAR T cells effectively killed the leukemic cells for the first time and second time. But these cells were less effective for the third round and lost their anti-tumor effect for maybe due to the immune exhaustions. As we expected, when we co-cultured leukemic cells with CD19 CAR T cells in the presence of ruxolitinib, ruxolitinib completely blocked the anti-tumor function of CD19 CAR T cells, probably because JAK1 inhibition impaired the activation of the downstream of the CD3 zeta chains. And interestingly, when we used the selective JAK2 pathway inhibitor, CD19 CAR T cell function was further enhanced even in the third round and prolonged anti-tumor efficacy. So we hypothesized that the inhibition of JAK2 pathway would prevent excess T-cell activation and the following exhaustions. So we collected the CD19 CAR T cells from the third round, as you can see here, and checked the expression profiles related to the immune activation and exhaustion. In a transcript analysis, we can see clear differences in the gene expression in the JAK2 inhibitor-treated CD19 CAR T cells, and gene set enrichment analysis revealed that the upregulation of the gene set related to of course, the JAK downstream, and the memory phenotype, and anti-apoptotic proteins, and ribosomal biogenesis that reflects the proliferation capacity of the T cells in the JAK inhibitor-treated CD19 CAR T cells. Then, we tested the synergistic effect of JAK2 inhibitor on CD19 CAR T cells using B-cell leukemia xenograft model. In this case, we used an in vivo stress test again and treated the tumor-bearing mice with a very low dose of CD19 CAR T cells in the presence of a JAK2 pathway inhibitor. As you can see here, the CD19 CAR T cells in the presence of JAK2 inhibitor controlled the tumor progressions, probably reflecting the immunomodulatory functions of JAK2 inhibitor on CD19 CAR T cells. So overall, the combination strategy of the CAR T cells with a molecular targeting drug would be an appealing strategy in the future. So in conclusions, the transposon CAR T cells have appealing characteristics as their simple manufacturing process and favorable memory-like phenotype. And the modification of the excess T-cell activation would prevent early T-cell exhaustions, leading to a prolonged anti-tumor efficacy. And obviously, further studies were warranted to maximize the clinical benefit of the transposon-based CAR T cells. Okay, thank you very much again. And all the idea and data I have shown today was our team's effort. And I would like to thank all the collaborators, including past members of our team. Thank you very much.